So this is today's agenda. We'll begin with some brief announcements. And our introductory trainee had some issues right now, so we'll actually start with our feature talk first. And then we will follow up with a trainee talk, and then the event will end. VanBug is a primarily student-driven organization aided by faculty members. I would like to thank Dr. William Shao, Dr. Amy Lee, and Dr. Faraz Hodge for their continuous support, as well as to the volunteers. So here are some tentative dates for the upcoming VanBug seminars. We will announce more details on our website. In the meantime, consider subscribing to our calendar and social media. We post update and seminar invites on these various platforms. We are currently in the midst of upgrading our mailing list, and that's why some of you who may have subscribed are not seeing any emails coming from us. We will post an update on that to the various platforms too when we're done transitioning. We invite speakers based on your suggestions. So if you have any speakers you're interested in hearing from, please visit our website and fill out the suggest a speaker form. VanBug is proudly sponsored by the Graduate Student Studies in Bioinformatics at the University of British Columbia and Simon Fraser University, Genome British Columbia, Langara College, and Stem Cell Technologies. VanBug is also affiliated with ISCB and our sister bugs, MomBug and TorBug. For the Zoom audience here, which is all of you, please ask your questions in chat and we will voice it for you. Our student talk will be presented by Madeline Eisminger. She's a master's student in bioinformatics at the Citgo and Sal Lab at SFU, and her talk is titled An Ontology-Based Approach for Tracking Viral Mutation Impacts. Our feature speaker of today is Dr. John Alkan, an associate professor at Bilkent University, and he'll be talking to us about acceleration of bioinformatics workloads through hardware and algorithm co-design. Can you take over?
Do I take over? Okay. Hi, everybody. Let me share. Okay. Hi, everybody. So first, I'd like to thank Jonathan and Faraz and the other organizers for inviting me. About 20 years ago, I was a visiting student at Simon Fraser University, so I actually attended VanBug as a student a few times. So today I'm going to talk about our work in the acceleration of bioinformatics workloads, and specifically I'm going to target the read mapping problem. Since most of you, I would assume most of you know how whole genome sequencing works. We have a test genome, and then we randomly share it into pieces. And then we take those pieces, and then depending on the specific technology that we are using, we can generate paired and short read sequencing, or single and long read sequencing, or in between linked read sequencing, which also generates short reads. And then we take those reads and then map to the reference genome that was generated by the Human Genome Project, or the more recent one from the telomere to telomere project. So this read mapping is actually a slow process. It takes a couple CPU months. So that is one CPU running in several months. But we can parallelize this to make it practically faster. But still, it uses a lot of the computational resources. So one of my students, Mohammed Alsar, looked into this and then tried to identify the bottlenecks in read mapping. So one thing that he found that 93% of the read mappers execution times is spent on the actual verification step. So this is trying to find the character matches and mismatches. We also found some other work which also showed that when we are using any kind of seed and extend mechanism, 98% of the candidate locations that come from the seeds are also dissimilar, because in the seeding step, we first find very short exact matches in the reads, and then we try to extend it using Smith-Waterman or its derivative. But 98% of the original seed locations actually fail to align. Another thing is that, again, with this, within the 93% from my first slide, when we are actually comparing two strings, the more sensitive algorithm, which is Smith-Waterman or its derivatives, has a worst-case quadratic time. So if you are comparing these two strings, for example, we have to compute this entire matrix, and then the edit distance is given at the bottom right corner. And here, when we are calculating this, there is also data dependency. So for example, in order to calculate this too, we have to calculate all the values to the left of it and to the above it, including its diagonals. Right? So this data dependency limit the chances of parallelizing this work. Alternatively, you can use WFA or other banded alignments, which does not necessitate to calculate this entire matrix, but they only work for high identity sequences. So therefore, if you would like to achieve higher sensitivity, then Smith-Waterman is the only way that you can do. So in the past years, I would say since 2013, we have been trying to make read mapping faster. We have been targeting two main approaches. One of them is filtering. So this is detecting the invalid mappings before actually doing the alignment in a way that is going to be much faster than the full alignment. And also accelerating the alignment itself. So for this, we can use Diddle-Movement, which is again the ancestor actually of Smith-Waterman. We also have the BiTAP algorithm alignment, and we also have the bit parallel alignment acceleration. So over the past years, we have used the SIMD instructions in the CPU. We have used GPGPUs. We have used FPGAs. And then more recently, we started to use processing in memory and also designing specialized hardware that is also processing in memory friendly. I'm just giving the names and the citations of some of them. I'm going to just give in more detail. I'm going to talk about the gatekeeper algorithm, which is actually an FPGA version of this SHD algorithm that we have developed earlier using the SIMD processors. So in 2017, we first developed an FPGA-based algorithm gatekeeper, which is a derivative of the SHD algorithm. And then we also extended it into two other algorithms, Shoji and Sneaky Snake, using again FPGAs and GPUs. Last year, we also published the GPU version of the gatekeeper, which also increased the accuracy of the algorithm. These are my students who have worked on this problem, and Mohammed is now an assistant professor at the Georgia State University, officially starting in January. So here is the idea. When we are calculating when we are comparing two reads or the reference or the query, we want to be able to achieve highly parallel matrix computation. So this is not an alignment matrix. This is a character by character similarity matrix. So this is similar to the dot plot analysis that was developed more than 30 years ago. So basically, when we have an A versus C, we are going to put a 1 because they are different, but A versus A is a 0 in this place because they are the same. And this is entirely data independent. So you don't have to, for example, to calculate this value, you don't have to look at any other value. So this is just this T versus this A. So therefore, we can just calculate this table in parallel without any data dependency. So if we had two sequences exactly the same, this yellow diagonal would all be 0. So, and when we have this stream of zeros, for example, the suffixes of both the query and the reference are exactly matching. So what we would like to do is actually starting from the top left corner, we want to find the longest stretch of zeros. So for example, if we just stay from here, start from here, which corresponds to this deletion of A, and then we just go along this diagonal, and then we switch to this diagonal, and then we continue, which is going to be a deletion of this A character, and then we are going to match everything else. So this is what we would like to do in general. So if we are looking for an edit distance threshold, which is E, and then we say that we are going to allow for at most 2 deletions or 2 insertions, then we extend this using a 2 deletion Hamming mask. So this is, again, the yellow is the diagonal, which is the exact matching position, or exact matching and mismatching positions. And then these 2 diagonals is going to correspond to 2 character deletions from the reference. And these 2 diagonals are going to be 2 insertion Hamming masks. So these are going to correspond to 2 character insertions to the reference. So what we need is basically this. We again still do not need the entire table. If we don't have any limitations, obviously we can just fill in this entire table, which is not going to hurt the data dependency problem, but it's going to cause us more computation. So the key idea of gatekeeper is therefore following. So if we have these 2 sequences, we generate, in this example, we are interested in 3 character deletions or 3 character insertions at most. So this is the original, this is the main diagonal, the Hamming mask corresponds to the main diagonal. And then we have 1 deletion, 2 deletion, 3 deletion Hamming masks, and then 1, 2, 3 insertion Hamming masks. So the optimal alignment is actually going to correspond to these green lines. Right? You can already see the stretches of zeros. This is what we would like to find, but obviously we are not going to just look at this large number of zeros and ones and now find this path. Actually, our other algorithm, Sneaky Snake, does that in a smarter way. So one thing we also do is, because these correspond to 1 character matches at every stage, right? There could be some spurious zeros in between. So for example, let me see. So there is just 1 character 0 in here, which we are not interested in, because if we are just coming from this 0 and then we are moving to this 0, we are actually doing 2 operations, 1 insertion and 1 deletion. So to limit that, when we see very short streaks of zeros, that is a single 0 or just 2 zeros, we just amend them and then make it 2 ones. So this actually increases our false accept rate, but it still saves us from any false reject rates. So when we have those, and then again, if we have this long stretch of zeros that we want, and then if we just do a bitwise AND on this, right? So again, these are the ones that I was talking about. And then if we just change those zeros to ones, this is the Hamming masks that we have. And then we calculate the AND. Right? So again, those long stretches of zeros are going to give you the ANDs. So this AND is going to summarize how many edits you still have to make. So at this position, everybody is 0. So everybody has 1, right? So all of those masks have 1. So therefore, you cannot really escape from doing any edit. It could be a substitution, or it could be a deletion or an insertion. Similarly in here, and then similarly in here. But for the other positions, we can already see the long stretches of 0. So therefore, there is a way that we are going to find these in the alignment. And after doing this, we just do a up count, that is the number of ones. So 1, 2, 3. So therefore, we say that we should do at least 3 edits to be able to align those sequences. So therefore, our estimated edit distance is 3. But if we just take out those two sequences and then we align them using the optimal algorithm Needleman-Wunsch, the actual edit distance is 4. So we are underestimating edit distance, which is still okay. So we are still doing some unnecessary computation during the alignment stage. But we are not overestimating edit distance, because if we overestimate edit distance, and then if we said the edit distance could be 5, and then if we just eliminate anything higher than 4, let's say, then we would miss this alignment, right? So it is guaranteed that we are not going to miss any real alignments, although we are going to still do a little bit of extra work. So therefore, just to compare the Needleman-Wunsch or the Smith-Waterman algorithm, there is data dependency. So this is the recurrence relation for the Needleman-Wunsch. And with the gatekeeper, there is no data dependency. Therefore, we can use independent vectors for each of those Hamming masks, right? And then we can process them in parallel using different hardware technologies. And all of those are very cheap bitwise operations, just XORs and then AND operations, right? So for this, again, previously we used the SIMD, which was the SHD work, which was then extended into FPGAs and GPGPU using the gatekeeper algorithm. And more recently, we have been using similar strategies, although not the same algorithm, using processing in memory. So speed is good. We actually get speed. I skipped the speed up results in this talk because the gatekeeper algorithm is actually not really new. I'm still explaining those to show what we can do and what we should do to achieve high parallelization. But how about the energy usage? So alignment takes time. No matter what computational resource you use, you are still using a lot of energy. So let's look at the cost of energy in clouds or in distributed systems, or even your single processors, your single servers. So this is a work from our collaborators that was published in 2018. And then they analyzed the Google workloads in consumer devices. So these are all the apps in your phones, right, that use Google services, basically. So what they realized was as following. So this is the traditional computer architecture today. So this is still what we have in our phones, in our servers, in our desktops, laptops. In von Neumann, so this is called the von Neumann architecture, and in the von Neumann architecture, the RAM and the CPU side are separated from each other. And then close to the CPU, we have 1 or 2 or 3 more caches. So this is level 1 cache, level 2, 3 cache. Some CPUs also have level 3 cache. But the real data always come from DRAM, move to those caches, and then goes to the CPU, processes, and then it goes back. So the data always comes back and forth, back and forth, back and forth. So these caches are fast, but the memory bus between the CPU and DRAM is not fast enough, and it uses a lot of energy. So one thing that they, our collaborators, Borman et al., realized that about 63% of the total system energy is spent on data movements. So between the DRAM and the CPU, you are wasting about 93% of all the electricity that you are using. Right? I didn't have this one here, but there's also another result that in Google Cloud Platform, just initializing variables. So all of you write codes, all of you in your code initialize something. They say integer counter initialized to 0. This is the initialization. About 5% of all the energy spent in the Google Cloud Platform is wasted into these initializations. Because again, you have to fetch the information from DRAM, set it to 0, and then move it back to the DRAM. Right. So one potential solution here is moving this computation closer to the data, and the data sits in the DRAM chip, right? We are not going to be able to put the entire CPU right next to the DRAM because there is space limitations, there is still energy limitations, but we are going to put some sort of a compute unit next to the DRAM, which corresponds to the processing in memory paradigm for computation. Right. So again, our challenge here is the limited area and the energy budget. You cannot put a Xeon processor right on the DRAM, but you can still have some simpler circuitry which can target some sort of specific instruction sets. So the processing in memory can be just thought of in two different camps. One of them is near memory, and there are some technologies that are currently available. I'm just showing two of them. One of them is the hybrid memory cube, and the other one is the DRAM processing unit from a company called Upman. So you can actually go and buy these. So this is commercially available. And what happens here is that this looks like a regular RAM chip, right? And now we have the memories here. And now right next to each memory chip, there is a small processing unit, the DRAM processing unit, DPU, which has direct access to DRAM. Since these are very small, right, and doesn't use much energy, you cannot do everything on those DPUs, but you can apply some of those instructions. Another way is, again, those chips, take those chips, and then instead of putting them next to each other, put them on top of each other, make this into a cube. And then between those chips, you also put high-speed transfer vias in here. So these are called the TSVs. So therefore, you can just access from one layer to another layer very, very quickly. And then at the bottom of this, you put another logic layer, right? So this is, again, some circuitry that has a direct access to the DRAM. So these are two different ways of near memory. Upman DPU is, again, commercially available. Hybrid memory cube, I believe Apple and Samsung are developing those. So this is in the prototype stage, so they are not commercially available yet. Another way of thinking this is using memory. So this actually includes a lot of electrical engineering. I cannot really explain what is going on here. I'm not an electrical engineer, but we have collaborators who work in this field. So this is modifying the circuitry within the memory, and then have the memory do some computations. There is another algorithm called Ambit from our collaborators, which uses the traditional memory chips and then modifies the voltages in those cells in such a way that it can do bulk operations. So bulk initialization, bulk AND operation, OR operation, XOR operation. So bulk bitwise operations. And by bulk, I mean at least on the order of 8,000 bits per operation. So they are all done simultaneously without needing any circuitry at all. So they just there is some additional circuitry that you need to play with the voltages in the banks. So this is another design from another of our collaborators, which again adds some additional circuitry to the memory, and then they use this to basically to detect viral sequences. So this is a K-mer-based virus detection algorithm, but this is entirely done in the hardware within the memory. So this is the using memory, and this is the near memory. So the near memory is easier to deal with, especially with the DPUs, because it gives you a programming paradigm very similar to GPU programming. So all you need is using the necessary SDKs, and then this actually becomes a parallel programming task, but this requires a lot of electrical engineering knowledge. So the first algorithm that we have developed using processing in memory, together with our collaborators at ETH Zurich, and then the first author was Jeremy, was published in 2018, so which is called the green filter. So the green filter, so this study was simulation-based. We didn't actually build any DRAM because building the DRAM takes a lot of money. It costs a lot of, it has a high cost. But looking at a 3D memory, again, these are the DRAM layers. And within the DRAM layers, there are banks, right? So this is true for any DRAM, current DRAM chip. But now we also have a 3D-dimensional structure. We use the structure of those banks. And then again, a bank is an array of cells with a row buffer to transfer data. This should come from your computer organization or computer architecture courses if you have taken any, which allows us to put bit vectors in banks, right? So what we do is we have a bunch of rows. And in the next slide, I'm going to explain this in more detail. And then each row is going to correspond to some certain k-mer. And then we can just put those bit vectors in those banks in such a way that they are easily accessible by the row buffer and across the DRAM layers as well. And here is the algorithm. So this is what I mean. So let's say this is our reference genome. And then we identify the size of a bin. So this is going to be a parameter. And then these are going to be overlapping. And then let's say we have these bins, right? So these parts are going to correspond to these bins. And then we are going to represent each bin with a bit vector. And within this bit vector, we have k-mers, which we also call tokens. And then we look at all possible k-mers in here. So this is just a simple way of demonstrating this. Let's say this is our bin 1. And then this is our k size, if our k is 5. This is all possible k-mers. And then we just say this AAAA, this is in bit 1. So therefore, this is 1. AAC is also in bin 1. So this is also 1. This is missing 0. This is missing 0. And then so on. So from bin 1, we generate one bit vector. From bin 2, we also, sorry, this is again just demonstration. And then we just generate these for all of those bit vectors, right? So if we have, let's say, k is equal to 5 and bins number of bins is 200, then the memory footprint is about 4 gigabytes. So we can play with those parameters to change our so this is for the entire human genome. This is the extra memory that we have to use in these banks. So this is about the presence and absence of each of those k-mers within each bin. Then, when there is a read coming in, and then we want to align this, and then we have from the seeding step, we identify which bin this might correspond to. So let's say this corresponds to one of those bins. And then we take the same k-mers, the same tokens, the same k-mers of size 5 from here. And then we look at the corresponding bit vector that is shown to us using our seeding algorithm, right? The initial seeding. And then let's say this is the bit vector that it corresponds to. And then we try to match those tokens to the bit vector. And then we just sum them up. I think I have a visibility error in here. So there should be k-mers in here. And then we sum them up. And then we compare with the threshold. So one thing here is that this does not look at the order of k-mers matching, right? So we have these two k-mers. So we have this k-mer matching. And then we have this k-mer matching. Right? Maybe this k-mer came, so this k-mer came before the second one, but in the bit vector they could be shuffled. Right? So within within each bin, the k-mers could be shuffled, so we cannot really prevent that. because we are just doing a Bayes-Kramer counts. And then we just compare it with the threshold. And if it passes the threshold, then we just send to the actual alignment stage for the alignment. But if the sufficient number of Kramers do not match between the read and the bin, then we just discard it out. So the properties are that the operations are simple. When we are given a bin, we are just looking at all the bits that correspond to each token in the read, which is similar to the pop count instruction. And then we only do a simple comparison against the threshold. So therefore, the operations are simple. It's also highly parallel. We can operate each bin independently. And there are, again, many bins. So they can all be interrogated at the same time in parallel. And it is also memory bound. So that is, all the data is in the memory. So we actually have to do a lot of frequent accesses to the memory. So therefore, instead of doing a lot of come and go between the memory and the CPU, if we can just do all of those operations on the memory, it gives us speed up. So therefore, these properties together make the green filter is a good algorithm for 3D stacked DRAM. So the next two examples, I'm going to just talk about very quickly, so I'm just going to mention them. We also developed an algorithm published in 2020 in the Micro Conference by Damla Şenol. And she developed an approximate string matching. So this is an alignment algorithm. And she developed a hardware framework for this. So if you do not know, the BiTAP is a pretty old algorithm. So it was initially developed for exact string search back in 1964. Right? And then in 1989, Bayes added support for mismatches and wildcards. So Bayes gave approximate string matching properties to BiTAP. Indas were later added into the algorithm. This algorithm from Member and Wu is actually the implementation of this is a tool called agrep, which is available in Unix systems. So this is approximate grep tool. Further improvements by Bayes and Navarro in 1996, and then in 2020, Damla added the capabilities of traceback because none of those had any traceback, so they just tell you whether they match or not. But it doesn't tell you where the reads do not match. So we now have the traceback. And then also reduced the data dependency in calculation, therefore providing acceleration. So therefore, this was 56 years in the making. So the BiTAP or the shift and methods, just very quickly, if you haven't taken any introduction to bioinformatics courses, works as follows. So let's say you have one text and one for. So this is the original algorithm. This is for exact sequence search. And if your pattern is for, you have 3 characters in the pattern. And you have California, you have 10 characters in the text. And then you again do a character by character match, but you don't do just the way that we do in Gatekeeper. Instead, you are going to calculate this M matrix, where if Mij is equal to 1, then the first I characters of P exactly matches the I character of T and you get character J. So, for example, in here, this for is here in text for, right? And then what we want, the first I characters, and this is exact match, and this is 3 characters, so therefore we are looking for 3 characters of match, right? In T, so this is in here. So if we have 1 in here, this means that we have a 3-character match from the pattern that ends in position 7 in text. Okay. So what we want is this one. But this one depends actually on this one, and this one actually depends on this one, which you can calculate column by column, which is done by using a bit shift. So what you are going to do is you are going to need a U vector from your pattern. So let's say here the pattern is ABAAC. So you're going to need 3 bit vectors, UA, UB, UC. And these are the positions in the pattern. So A is in position 1, 3, and 5. 1, 3, and 5. B is in only position 2. C is only in position 3. So you just pre-calculate this from the pattern. And then a bit shift operation is basically just take a column, shift it by 1. Right? So you just shift. So this 0 goes here, this one goes here, this one goes here, this 0 goes here. This one is lost. And then you set the first bit to 1 always. Okay, so this is a guarantee. And then what you do is you're going to do this column by column. You just take this one, you bit shift it, and then you end it with the first, with the U vector that corresponds to the first character, for example. And then you continue, and then you have a match. Right? So this is lots of just shifts and AND operations. Again, bitwise operations, but there is some data dependency here. And again, this one does not give you any traceback or any indels or mismatches. But our algorithms, along with Bayes' algorithms, provide these functionalities. So the GenAsm was the first BiTAP-based or the shift-and-based read alignment approach. So again, this doesn't do any filtering. Instead, it calculates the alignment itself. And it was also optimized for both short and long reads. And the idea is just a simple idea is that we have the memory. We just take the reference and the text places, and then the reference and the reference text and the query patterns. And then the CPU pre-calculates the seed locations, and then everything is passed into the accelerator. So this is the distance calculation accelerator, DC. And TB is the traceback accelerator. So first, the distance is calculated. Then if the distance is lower than some certain threshold, it is passed into the traceback. I'm going to skip these details because it could be just too much, but we do have some circuitry that does this. So this is a co-design of a modified pin-friendly BiTAP algorithm and the hardware accelerator. So in this study, we modified the BiTAP algorithm to enable efficient alignment of longer patterns, remove the data dependency between iterations, therefore providing the parallelism of the amount of iterations. And if you are more interested in this, you can go to this YouTube link where Damla actually talks in detail about this algorithm. Another algorithm that she also developed was Segram. So this is again a hardware accelerator. For this time, it is for sequence to graph. It also does sequence to sequence mapping, but also does sequence to graph. So sequence to graph mapping is as follows. So traditional what we have been doing in the past many years is that we have a linear reference, and then we just compare each read with one single linear reference. So this gives us something called a reference bias. So because now we cannot really differentiate between the read sequencing errors in the read and the real variation. So therefore, it is more likely that we are going to align the reads that are more similar to the reference sequence than the ones that are not. But if you are looking for real variant, obviously, those are going to be mismatches or indels in the read, and our alignment algorithm may miss those. So to address those challenges, an alternative approach can be used, which is using a graph-based reference. And then let's say this is our original linear reference, and then we have already identified some variation in 3 different individuals, let's say, in this cartoon example. And these are the differences. And then we just model this into a graph. And here, all of those have the same ACG in the beginning, right? So the linear reference has a T after this ACG. This individual has a G, which is shown with this green. This individual has T and T, so that is T and T. So we have T and T, both of them. And this one is missing a T. So this we are just going to skip that one. So this is the skipping, right? So this is skipping. And then we can just build this graph. which represents all of those variation, the real known variation, into a graph structure. So now our problem becomes aligning the reads into this graph, right? But it is a more difficult computational problem with, again, no prior hardware design. So Segram provided this. Again, it is composed of two different accelerators. One of them is for the seeding. So this is, again, quickly finding the nodes or the edges that correspond to short subsequences, and then extend the alignment by moving on the graph, which corresponds to the bit-aligned accelerator. Again, I'm going to skip the details, but just as a result summary, if we just use the sequence-to-graph mapping with the accelerator, we can see from 6-fold to 100-fold speed up. So this is for short reads, this is for long reads, compared to graph aligner. And also using about 4 or 3 times less power than graph aligner, which is, again, obviously a CPU-based graph aligner algorithm. And then similar numbers corresponding when we are comparing with VG, which is the most used graph aligner today. So this is for the CPU algorithms. There is another tool called Pascal that also does alignments, although it is not a specific hardware implementation. It uses the CPU's AVX512 instruction set. So this is SIMD instruction over the CPU, but with the SIMD instruction. Again, we see a good speed up. We also have some more modest improvements for sequence-to-sequence alignments. Again, the details are available on YouTube. So this is Damla's talk from the HiTSeq Conference in 2022. So this brings us to our current project that is funded by the Horizon Europe program called BioPIM. So this project actually came out of boredom during the pandemic. We were already working in processing memory technologies, but we were doing it for free, let's say, because we didn't have any funding specifically for this project. But during the pandemic, since I was bored at home, I started emailing friends, and they started emailing their friends, and within a day or two, we had our consortium. I guess everybody was bored at home. So we brought together Bilkent, so this is my university, ETH Zurich, Institut Pasteur in France, CNRS in France. Upman is a company in France that develops those DRAM processing units. IBM Research in Zurich, and Technion and Barilla universities in Israel. So this project started in 2022, so this is over 2 years now, which targets in-memory alignment search algorithms, global and local assemblies, pan-genome analysis, so this is the graph mapping, and also variation calling and base calling. We are developing accelerators using PIM, which gives us cost, energy, and time savings. This is just the overview of our project. We start by doing some workload analysis. So this is, we just take the traditionally used algorithms such as BWA, Minimap to Canoe, and then we analyze if they are memory bound or not. And then we decide if we should target them with any processing in memory. And if we should, which one? Is it going to be the near memory designs or the using memory designs? And then depending on that, we develop the necessary acceleration, and then we integrate into software. Right? So this is the main flow of the BioPIM algorithm. So I'm just going to show a couple of examples. Most aligning tools are memory bound. So this is the memory boundness of the BWA, for example. So memory bounds is about 40% memory bound. With graph mappers, graph aligner is memory bound. So this is the amount of the percentage of time spent in memory versus in the core. But mini graph is, for example, not memory bound. Mesh map memory boundless analysis in here, and with the long read sequence mappers, LRA was the most memory bound, but for Minimap2 and NGMLR, it was not as bad. So one of my students, Ömer, is working on the workload characterization and then also the acceleration of the graph mapping. So this is for graph mapping. And then he also analyzed function by function, which functions are used. So this is graph Smith-Waterman algorithm using the SSE2 SIMD instruction set, basically. So this is about 97% memory bound. This comes from VG, actually. So these are what we are going to target. So now Ömer is working on a design called SegaPIM, which uses the UpMem DPUs, DRAM processing units, to accelerate the sequence to graph mapping, which necessitates actually partitioning the graphs into DRAM chips, because one shortcoming of the current generation of UpMem DPUs is that these DRAM chips do not communicate with each other. Right? So once you put some of your data into the DRAM, into a chip in the UpMem, it stays there. So you cannot communicate between those two chips in the current iteration, but UpMem is going to release soon a new design which enables communication between those different PIM chips as well. So some of the other samples from our collaborators, CNRS, so this is a French group based in Rennes. They accelerated the Needle-Munich algorithm using the UpMem DPUs. So this is the speed up they get, right? So about 9-fold speed up for ribosomal RNA alignment, but about 8-fold speed up for PacBio long read alignments. So I'm just going to give very quick summaries because I think I am close to the end of my time. They also implemented Bloom filters in DPUs, which gives more modest speed up, so only 2 to 3-fold speed up in UpMem DRAMs. So this one reason for this is again because there is no communication between different PIM chips in the current iteration. ETH is doing something. ETH is actually doing a lot of different things. They are developing, I already mentioned Mbit, which is using DRAM. This is modifying the voltages. They are also developing a tool called GenStore. So this is processing in storage. So this is direct computation on the SSD drives, right? Because SSD drives also have controllers. So they are using them to do some computation. And they have an extension to GenStore called Megis. It will be published soon. I believe it is accepted to a conference, which uses the GenStore strategy for microbial for metagenomic analysis. And then they also have this called Venice. So this is near the router. So this is the processing is done on the network router. So when you are downloading the data, it is processing it on the router before even it reaches your computer. Barilan is developing an algorithm called GAPIM, which is an acceleration of the pair HMM within the GATK software package for variation discovery. So they achieve some acceleration, although it is modest. And Institut Pasteur is actually developing, among other things, they are developing an API called the BioPIM library. So this is currently available. It is under development. It's not complete, but it provides an API in C++ that makes DPU programming easier. So here is just an example. So this is an example checksum calculation using UpMem. All you do is just you define your what is going to be done in your launcher. So this is the checksum operation. Right? So this is what you're going to do in the checksum. Let's see, this is simple C++ code. And then you just use the BPI library, tell the launcher on UpMem, and then launch using 200 DPUs, and you just get the result. Right? So all of those is distributed into UpMem. And then actually, this also provides another parameter here. You can just switch this to another keyword. I don't remember the keyword, but it can just parallelize onto the regular CPUs as well. So you can use this as a easy way to debug your code before using the DPUs as well. So finally, I would like to thank many students in ETH, Carnegie Mellon, and Bilkent. And this is my group, my current group in Bilkent University, and also our Bioinformatics consortium. I just put the names of the team leaders, and some of those are in here, and our collaborators and our founders as well. Thank you very much. Okay.
Thank you very much. Thank you for accommodating and speaking first. Fortunate circumstance.
I mean, it is better for me because it is already 11 p.m. here, so I don't want to wait anymore.
So while people might be asking, I do have some questions I'd like to ask you. So for your rim filter, you mentioned there's like a threshold, I believe slide 18. So I'm just curious, how does that work with like a long read? Because you would expect there to be a very high hit for pretty much every read, right? So is there a different threshold here, or?
So yes, you're going to adjust the threshold based on the read sequence. Obviously depending on how. So this threshold basically just says how many k-mers within your read hit this bit vector, right? So if you have a long read, obviously this threshold has to be higher because you are going to have more k-mers already. So the higher number of k-mers, the higher number of matches that should be in your set.
Is that automatically detected, or is that the parameter you set?
It is no, it is automatically detected. I believe there is a parameter for the maximum edit distance, so based on the maximum edit distance, it detects the threshold itself.
Okay. So another question on this one, I believe it's the next slide. So the number of bins is essentially sorry, previous one.
Okay.
Yeah, so you said 200 bins, right? So that's dividing your genome into 200 separate bins.
Yes.
So for a human, that would mean each bin is give or take 15 megs. How does K5 work? Because that's what 1,000 something.
So here is a trick. Here is a trick. Once first you can process each chromosome separately. So we don't do 3 billion divided into 200 bits. So it is in an iteration, right? So for chromosome 1, it is 250 million characters. Then you divide it into 200 bits. Then you process it. Then you take the next. Then you take the next. So that is one trick that we didn't mention here. And I believe, okay, I don't remember this one much. So this is from 6 years ago, actually. But it also partitions chromosomes in a way too, I think, as far as I remember.
I see. Okay, that makes more sense. And sorry, just the last quick question. So how easy do you think it is for like existing infrastructure to transition into the BioPIM architecture? Because the speed up there is substantial, right? And it would help with a lot of applications, but transitioning is not easy. I don't think. So yeah, I want to hear your take on it.
I know. So the transitioning is not easy. So one good thing is that UpNAM chips, as far as I know, they are not expensive. And they are readily available. And the libraries that we are developing is making it easier. So about 2 weeks ago, we had another meeting, and then so it was very, very new. So that's why it's not in the slides. But we already have some tools implemented in UpMap DPUs, and these are the tools that you always use. So for example, metagenomic classification, there's an algorithm called MASH. There's a mean hash-based metagenome distance estimator. So it is ready that you can just download and use. Some other tools are under development. We have 2 more years in this project, so before we are done, we would like to develop many more, we would like to port many more tools into the DPU structure. But we're also providing the library. So if you already have your code written in C++, and if you are already doing some sort of OpenMP-based parallelization, it is easy to convert into Biopim library.
Okay, so we have a question from the audience with regards to Apple chips, basically. So how does Apple's SiP chips M1 to M4 with integrated memory and high memory bandwidth up to 800 gigabytes per second help solve some of these problems with hardware architecture, speed up, and energy use?
Okay, I do not know about Apple's SiP chips. But high memory bandwidth, okay. I do not know about these chips, but the high memory bandwidth is going to be useful because if we are moving data, even if we are moving data within the memory, then it's going to give us some speed up. Again, I'm not really familiar about this chip. Sorry. Okay, so I see another question. Would you like to read it or?
Oh, it's just
Okay.
Do you say, okay, I'll read it. Do you think the future sequencing centers will need to have PIM modules and FPGA cards as part of their standard compute infrastructure?
So FPGA cards, so that's an easy answer because if you are familiar with the Dragon platform, so this is from Illumina. Dragon is an FPGA accelerator, so which is embedded into the Illumina sequencer itself. So it is already part of, yes, they have Dragon FPGA compute nodes. So Illumina is already investing in the FPGA with Dragon. So it will be standard because Illumina is going to make it standard. So for PacBio and Nanopore, I am not sure what is the current situation. But you still do not need to invest into FPGA because FPGA nodes are available in Amazon and Google Cloud as well. So for the PIM modules, we do hope that they will be available in the cloud platforms as well. But investing in them locally, I am not sure. But since they are going to be much cheaper than FPGAs, by the way, PIM modules are much cheaper than FPGAs. It's going to be more straightforward to have them in-house. But FPGA is already in your Dragon. It's already in your clouds.
Can I ask you questions? Okay, I have a question, John. So looking at the earlier work, you were just showing for dropping out the bad candidates, right? So you basically try not to do the alignment, but just rather filter out as much as you can without losing the sensitivity. So just in ballpark, right? What is the amount you drop, right? I mean, you should have a statistic, right? So you do, based on this calculation, you drop a lot. You still want to keep some, which is going to be still wrong alignments. So what is the drop? I mean, that's the biggest improvement. You're trying to get the quadratic down as much as you can.
Yes, it could be in my backup slide somewhere. So I believe, okay, let me see. I think this is the relevant backup slide. So yeah, this is pretty much the number. So from the 10 to the 12, you're dropping to 10 to the 3. So this is basically a ratio. This
9
is
order?
Yeah, about on the order of 9.
Okay. And
I think that would be my backup.
that's significant. And the other one, again, following up on Jonathan's question on slide 18, I still have a little bit of a hard time understanding. So I understand the bins and I understand what you're trying to achieve here, but what happens when you have low complexity regions? Right? You're going to hit the low complexity regions.
Oh yeah, we are going to hit them.
It's going to be causing you a lot of issues.
It is going to, it's going to, it won't be able to filter those out. You are still going to try to align them.
Yes.
So yes, it's going to cause what we call false accept rates. It's going to increase the false accept rates. So you're still going to try to align them. So yes, that will happen. But we are not going to miss anything that we should align.
Yes. And what's the effect of not having an order here? Like, I mean, you can imagine you can have like a rearranged region, which probably would be perfect. Like, how often that's going to be increasing your false accept rate as well? I mean, you have low complexity, and you also have no order here. You're just looking at the composition.
Yes. Yeah. So the no order is going to still cause problem in the low complexity. But if you are if it's not a low complexity, so it's not a high, it's not a low complexity region. If it is more like random looking two strings, right? Then the k-mer composition is not going to affect much. So that you're not going to see a lot of shuffles between k-mers. If the identity is very low between two strings, their k-mer composition is also going to be very low. So this is going to be very similar to comparing documents with min hash, right? So it is essentially what it is. I mean, so more implicitly, this is kind of like min hash. You are looking at the intersecting k-mers versus non-intersecting k-mers, which is done in min hash and for document comparisons as well.
Just one final question, and I think Ibrahim will be chiming in probably. I mean, is there a plan providing libraries, right? As we, the community and students, we would like to implement like the libraries. We don't want to just get into details of understanding any of these in detail, like the architectural things, because that's a lot of optimization. Will be libraries that is going to be, I know that you're just trying to do for C++, right? But the trend has been changing significantly. So is it going to be for high level languages? I would say Python.
Python, no, as long as I am alive, it's not going to happen. Currently, C++ is the way that we are going, but there are some plans, especially from the UpMem side, to provide Rust support. So for C++ and Rust, there will be. So from the Pasteur side, so Institute Pasteur is developing C++, but UpMem has Rust in their list. For Python, not really, that's not our plan. But if this is Ibrahim Nomanagic, maybe he will be interested in integrating into his Python-like programming language. Was that Ibrahim Nomanagic you were talking about? Okay. So yeah, it is C++ and Rust are in the class currently, not Python.
Okay, if there are no other questions, thanks once again for speaking at the seminar. And we will move to our trainee speaker. Madeline, are you ready?
Thank you. I'm just stopping here. Okay, thank you, guys. It is past 11 p.m. here, so I'd like to say goodbye, good night for me.
Right.
Thanks for having me again. Bye.
Okay. All right. Thanks for switching the talks around. Can you all see and hear this okay?
Yes.
Yes. Okay. Okay, thanks for the introduction. I'm Madeline, like Jonathan said, and today I get to talk about, very quickly, just about using ontology models in the context of viral surveillance, which is part of my master's project. So to lay the groundwork, I'm gonna remind you of a thing that happened a few years ago. Viruses are all continuously changing. We know this about SARS-CoV-2 from reading the news cycle, but it's true for all viruses, RSV and influenza and Ebola and everything. Viral evolution, as we know, can impact viral transmission, diagnostics, drug and vaccine efficacy, and disease severity, among other things. This is an example from the New York Times. It's a headline saying mutation allows coronavirus to infect more cells. Study finds. Scientists urge caution. Yeah. Yeah, so some of these changes at the molecular level don't make much difference at all in how a virus behaves, and some mutations might change it a lot. So maybe like this, a new strain of virus is more transmissible, maybe it evades immune system defenses a bit more easily, or maybe a drug that worked well to treat last month's version of the virus doesn't work so well on this new one anymore. The Delta variant, the Omicron variant, and others for SARS-CoV-2 are all examples of viral strains that have significantly different behavior than the ones that came before. In BC, we keep track of viral genome mutations in a few different ways. The BC CDC monitors evolution of a bunch of respiratory viruses, RSV, SARS-CoV-2, influenza A, and influenza B through community-wide testing, which you'll remember, and through wastewater testing. So a couple times a week, wastewater samples go to the BC CDC, and they figure out what lineages are floating around today or this week compared to last week. Across Canada, SARS-CoV-2 sequences are all uploaded to the VirusSeq data portal, and from there, my colleague Zohaib has developed a workflow called Virus MVP that calls and visualizes viral mutations from that database. And the last time I checked, there's more now, but last time I checked, there have been around 18,000 SARS-CoV-2 mutations found in Canada so far, and the genome is only about 30,000 kilobases long. Or, sorry, 30,000 bases long. So we know which mutations are around almost in real time, but it's also important to know what those mutations mean for us in real time. So how do we get that information? And if we know what the latest mutations are, and we know what those mutations typically cause, then we can hopefully predict how a new strain will behave, and then we can respond well to make sure that people get the drugs and the vaccines and the public health measures that are right for them. And this is the tricky spot. This is where we hit a bottleneck. So mutations, those we can find, but getting information on the mutation impact is a bit trickier. There's no standardized resource for this so far, and finding research on it is pretty hard work. If you want to do it manually, there's hundreds of thousands of publications for you to sift through, and you have to guess the research terms for them. Just in the beginning, a few months of the pandemic in 2020, around 87,000 publications about COVID-19 came out. All these publications use different terms for the same mutation. There's a bunch of different naming formats there. And they all use different kinds of phrases for the same kinds of mutation impacts. There's lots of different ways to say transmissibility or virulence or all of those other things. And without a shared language, it's really hard to share data quickly in a meaningful and standardized way. So we need a standardized vocabulary and a database that links viral mutations for viruses just in general to research about what those mutations mean. This is where ontology comes in. In case you haven't heard of these, ontology is a philosophy thing, but it's also a computer science thing. An ontology is a standardized way to model knowledge. It's a machine-readable knowledge web made up of nodes and edges. So in this tiny, tiny model over here, the nodes are boxes, and the edges are the arrows here. Blue means ISA, and the one that says ribosomal translates to means ribosomal translates to. Each node and edge has a label, for example, chemical entity, universal resource identifier, so we can always find that same thing, for example, KEBI, and then the number 24431, and it also comes with a definition. This kind of knowledge web will be the backbone of our database, and it gives us a structure to query over and reason over, so we can use this model to pull out different mutations that correspond to an impact, or we can find all mutations that find all mutations in the reverse direction, or anything we want, depending on what goes in here. In this very, very simple example, this is just a tiny, tiny ontology model. A sequence mutation is a chemical entity, and there's two kinds of sequence mutation. You can have nucleotide sequence mutation or an amino acid sequence mutation. And we know that a nucleotide, when ribosomally, translates to an amino acid. So this is just us telling the computer about what a mutation is in the context of different kinds of sequences. In order to make this database, we need some data to start out with. So the most comprehensive list of mutation impacts on the web so far that I've been able to find is called POCE, and it's been created by Dr. Paul Gordon at the University of Calgary. It's a GitHub repository, and he's gone together with his student Kushi have manually been sorting through the literature and dividing into 43 different functional effect categories. This purple one shows like pharmaceutical effectiveness, and all the text files have different names. It includes 326 evidence sources, last I counted, and covers 11,000 mutations. Right now, it's based on SARS-CoV-2 because that's what we have the most data for, but it's expanding to Mpox, and soon enough, there'll be flu in it too. If you open up that one pharmaceutical effectiveness file, you see a bunch of records inside. There's this human readable description about the mutation effects found in the paper. There's the paper we found it in, and there's a mutation named there. So these are the kinds of things that we want in our ontology model later on. And the first thing to do is to come up with standardized terms for all those impacts, those 43. The details, I think I'll go quickly. So start with the POCE categories. We read through every single file and every single paper and come up with, really read about what's in them, what's been reported, what kind of public health interest there is, what kind of phrases they use in the paper, look at those all together, see what terms already exist in the ontology world, make a suggestion, and discuss with experts until we come to a conclusion. This is a small slice of what happens in the end. As you can see, what used to be pharmaceutical effectiveness is now called drug susceptibility of infectious agent. and it's a term with IDO number. This hierarchy of impacts is virus agnostic, so it can be used for other things like mpox or RSV, and it's structured in such a way that it should be easy to add new terms as they come up or as we need. And it's able to capture for each research study which impacts were directly measured in an assay, as well as which impacts were maybe inferred or concluded from that assay, as well as which ones correspond to a specific part of the viral life cycle. There are subclasses underneath, so we can say how something has changed based on mutation. Has it been increased or decreased? Do we not know? Has it just been altered? If the mutation is about the virus becoming more susceptible to a vaccine or a drug, we have another tag for the vaccine or the drug that goes with it. And then we can specify if it means resistance or if susceptibility has been increased or decreased. Where are we? Yeah, so once we have that functional impact model, we can put it into this general evidence model, which covers the very most essential information, which is recorded in POCA and other mutation impact resources. Just like we saw in POCA, there's a mutation functional effect record that has some different parts. It's got that description, it's got at least one effect, and that effect is positively correlated with some kind of sequence mutation symbol, which also has a standardized form now. And it mentions the reference document, so that's the paper citation of where it came from. The green impact model is what we just looked at, and there's two other models hidden in here. It's the mutation model that puts mutations in context of genes and viruses and proteins and reference sequences, and the literature model, which gives them a broader citation. If we zoom in on here's a worked example, so less general one. This is a paper by Barton et al. In 2021. And Barton et al. Found that N501Y, a mutation in the spike protein of SARS-CoV-2, is positively correlated with increased binding affinity, specifically binding a spike to the ACE2 protein in humans. So this is the ontology way to write it out. We can write the description with this has value box, and that's just some text that's nice and easy for a human to read. And we can also have it as the subject predicate object triple, which is searchable by a computer. This is when, so now that we have this mutation impacts model, we can use it for a few different outputs. So mutation impacts here are overlaid on virus MVP. Virus MVP is a heat map visualization of viral mutations here with their functions on top. And finally, I put it back here, but finally, this ontology framework has been captured in a data harmonizer template developed by Ivan and Damien and Kenneth in my lab, and a contextual data specification. It's available on GitHub, and this template is a spreadsheet style thing that provides a standardized way for researchers to easily record viral mutation impacts as they come across them or as they're in their papers. And then they can upload this to the future database from there or to POCA, or it can be used to overlay custom functional annotation data onto the virus MVP visualization themselves. And I have it over here. I can just look very quickly. So Haib and I have made this based on software developed in my lab already. So you can, there's a SARS-CoV-2 one and an mpox one, and there will be more to show up. But you can input your data in here. Some of them are given already. But for every mutation, we require a nucleotide position and a name, and then a standardized name for amino acid. Genes, gene symbols, protein name, protein symbol are all dropped down and given for you. So the names have to be all the same. And then over here is the variant functional effect part we were just mentioning. So for every mutation, you can choose as many as many mutation functional effects as you can, and it's split into measured variant functional effects, which is what you actually see in your assay. Inferred variant functional effect is what you didn't directly measure, but based on contextual knowledge, you can infer. For example, if you're measuring ACE2 binding affinity in some assay, you might infer that increased ACE2 binding means increased transmissibility for this new virus with this mutation that you have. And the life cycle functional effect is borrowed from the gene ontology and includes different, there's 6 or 7 parts of the viral life cycle that are implicated in this mutation effect. Finally, longer description, some identifiers that might be useful, and a place to put in a citation, and your name at the end. Yeah, so in the end, we have the ingredients for a database upcoming that links viral mutations to their impacts found in the literature, which will hopefully be a useful resource for researchers to upload to public health authorities to use, and just in general to make data sharing about viral impact mutations a lot easier and more streamlined in the future. Thanks to all these people. On the left, coworkers from Simcoe, and on the right, Koshi and Paul at the University of Calgary for their work with POCA. Thank you for listening.
Thank you for your presentation. We're now fielding questions. If there are no questions, then this is the end of this month's VanBug seminar. Thanks for coming.